our next speaker would be Mariana Braga. She has a PhD in animal ecology at Stockholm University, which she started working on evolution of butterfly-plant interactions. Then she did a postdoc at Washington University in St. Louis, where she mainly worked on method development. She did a second postdoc in Sweden and Finland, and from January 2025, she'll be an assistant professor at the Swedish University of Agricultural Sciences. So thank you very much for accepting our invitation, and the floor is yours.
Thank you so much.
Thank you.
All right. So, hi, I'm Mariana. Thank you so much for this invitation. It's a pleasure being here. Yeah, I'm very happy to talk about, I guess, one of my favorite topics, which is reconstructing evolution of species interactions. And yeah, more specifically, I'm going to talk about post-repertoire evolution. So just to make sure that, yes, okay. So I'm pretty sure everyone here appreciates, of course, the importance of species interactions. Every species interacts with many other species, providing or receiving food, protection, dispersal. And these interactions form ecological networks like the ones depicted here. And we have learned a lot about ecological networks in the past few decades, but there's still a lot that we don't know. And one question that I am currently trying to answer is, do we know enough to predict new interactions? That would be really useful in many ways, helping us assess positive effects of new interactions, such as species persistence, ecosystem functioning, chance of success of ecological restoration, and also some negative effects, such as those caused by invasive species and emerging diseases. And to give one concrete example, let's take one everyone can relate to, mammals and coronaviruses. So even though each virus strain is fairly specialized to its host species, they probe new hosts whenever they get the chance. And sometimes it works. They find a new host where they can establish a viable population. And when the novel host is us, humans, we call it an emerging disease. And it would be great if we could assign probabilities to these events. And to do that, we need to understand how this process of forming new interactions works. The kind of ecological interaction that I study are parasitic interactions, which are intimate, antagonistic interactions, for example, between a butterfly caterpillar and their host plants, or a plasmodium and birds, or tapeworms and fish. So maybe not everybody is used to the idea of thinking about herbivorous insects as parasites, but they are very much so behaving like parasites when they are caterpillars. They completely depend on usually a single individual host plant. So in this paper from 2018, we show that even though these three example systems look quite different, they share many similarities, which suggests that similar evolutionary processes may have shaped their interactions. And in order to investigate these processes, we need two types of data. We need phylogenetic trees for both interacting clades, as in this cartoon. And then for each pair of species, we want to know if they interact. The ideal scenario would be to know which pairs do and don't interact, and which can potentially interact. Because in order for a parasite to colonize a new host species, it must already have some potential to survive on that species. Otherwise, it will die before getting any chance to adapt to the new host. So distinguishing between the zeros and ones so the no host and potential hosts, is a crucial step because they don't have the same probability of turning into twos, actual interactions. To get this kind of data, though, one can do establishment tests in the lab. Luckily for butterflies, all you have to do is Google and follow the instructions from blog posts like this one. But jokes apart, you can actually rear caterpillars in the lab, give them different plants to eat, and see which ones work and which don't. So my PhD supervisor has done that about 20 years ago with a few butterfly species from the same clade, and that's a precious data set. I don't know many studies like that because it's a lot of work and it's hard to scale up, usually being restricted to individuals from the same local population and a few host species. But there are, at least within Lepidoptera, there are a few really precious data sets like that. In reality, the main source of interaction data for parasites are species descriptions. The problem is that you only get the one host and locality where the parasite was first found, but for many clades, that's the only available data. There are studies like this one, where many fish species in a given place were sampled and checked for a given parasite genus. And that way, they could quantify the strength of interaction between each species instead of using presence-absence and combine it with phylogenetic trees. The main source of interaction data are books and databases that compile many observations of ecological interactions. The problem here is that there's often a mix of good quality and bad quality data, and usually some false positives. But they can cover a whole continent or even the whole world, which is great for macroevolution and macroecology studies. So each kind of data has its advantages and disadvantages, but this is the kind of data that I have used the most, curated data from the literature. So butterflies and their host plants have been used as model systems for development of a few hypotheses of how interactions evolve and how that can lead to parasite diversity. The two main hypotheses are the escape and radiate from Ehrlich and Raven and the oscillation hypothesis from Jans and Nylin. And they are not mutually exclusive, as we'll see, but they focus on different processes. Escape and radiate focuses on the role of new interactions allowed by key innovations, while the oscillation hypothesis focuses on the ability to use hosts that were used in the evolutionary past, thanks to a genetic memory that is retained through time. So in this paper from 2018, we simulated network evolution and showed that the evolutionary processes in each hypothesis produce different network structures. The escape and radiate produces modularity, while the oscillation hypothesis produces nestedness. And when we looked at empirical data sets, they showed signs that both processes have shaped present-day butterfly networks. So that meant that we couldn't simply use network structure to explain diversification in these groups. And then we had to propose a unified evolutionary process for how hosts can drive parasite diversity, combining both radiations and oscillation in host range. But then to directly test that, we would need some sort of time machine, because unfortunately, fossil records of species interactions are very rare and non-existent for butterflies. So that's why we developed a model of host repertoire evolution for Bayesian inference of ancestral host parasite interactions. And to put this model into context, so here's a table from this review we published a couple years ago. where we group these different methods to reconstruct different aspects of coevolutionary history into categories, 3 categories. So pattern-based statistics, they decompose the host and symbiont phylogeny into phylogenetic distance matrices to test the extent to which the interactions could have been produced due to chance alone. So we're quantifying cophylogenetic signal. Event scoring methods, they map a symbiont phylogeny into a host phylogeny using the classic cophylogenetic events. So co-speciation, host switching, duplication, and so on. And each of these events is assigned a cost to determine the lowest cost mapping. So these methods can be used to test whether diversification in your group is better explained by co-speciation or host switching speciation, for example. And then generative model-based methods, which is the category in which the host repertoire model is in. They use probabilistic models to describe the process that produce observed cophylogenetic patterns. And with those methods, we can infer the probability of ancestral states and events. So when we started developing the host repertoire model, we knew we wanted to resolve common problems with other available methods. And most of these problems had to do with how people coded host use as a trait. Some methods only allowed one host per parasite taxon. So the tip in the tree, it could be a species, could be a genus. So either you had to focus on one interaction or repeat the analysis for each host taxon, doing one at a time. But then we are assuming that interactions with different hosts evolve independently. And we know that that's not true. And the third approach was to look at how the number of hosts, so the host range, changed over time. But then you're completely ignoring the identity of the hosts. So we proposed host repertoire as a better way to code host use because it includes both the information of which and how many hosts are being used. We represented host repertoires as a vector where each element represents a host taxon. So age 1 is a host, age 2 is another host. And each one of these elements can have one of 3 states, 0, 1, or 2. 0 means no non-host. 1 means potential host, and 2 means actual host. So actual hosts are those that you can observe that interaction in nature. And potential hosts, you can interpret differently depending on your system. For butterflies, the easiest way for us is to think about hosts that are not used in nature, but if you give that plant to the caterpillar, it has the ability to eat and survive on that host plant. So they already have some of the abilities necessary to interact with that host. But it's not quite complete yet because for some reason they don't use it in nature. And yes, and then the transitions between these states, so how they change over time. In this model, we have a 2-step process for both gaining new hosts and then also for losing hosts. So we don't allow an instantaneous change from 0 to 2. It has to always go through the state 1. And one cool thing about this setup is that you can easily remove the state 1, and then you have a 2-state model with only 0s and 2s. Then it's more of a presence-absence type of model. So this model is implemented in RevBase, which is a software for phylogenetics. And then the main features of this model is that the species can use multiple hosts at any given time. So both in your data and also in the ancestral states. We can include potential hosts, so the state 1. And we can also infer whether the phylogenetic proximity between hosts affects the probability of gaining new hosts. So here we have a hypothetical parasite tree, the big gray one, and then we have a hypothetical host tree in the bottom, and extant interactions in the top. So the data in the top, the repertoires here at the top, they are our observed interactions. And based on these three informations, what the model is going to do is to infer the root state, the root state. So which host was the ancestor of all of these butterflies or all of these parasites using? And then how does that repertoire changed over time along the parasite tree, producing the observed states? So, and here these blue arrows represent host gains and the red arrows represent host losses. And then when we do the reconstruction of ancestral states, we can either look at the internal nodes of the parasite tree and ask which hosts were they interacting with, or we can look at different points in time because we have a time-calibrated parasite tree. So then you can choose arbitrary points in time in the past, ask what were the lineages alive at that point, and then what were their host repertoires. So so again, this is the paper where we propose this model, and this is the empirical example that we show in this paper. So here, if you see my mouse, here on the left, there's the butterfly phylogeny for this butterfly tribe called the Niphalini. And here in the bottom, we have a phylogenetic tree for angiosperm families. So here we have butterfly species, plant family interactions in this matrix over here, where each square represents an interaction. And then these small squares represent these potential hosts. Is that data set from my supervisor that I told you about before. So these are tests that were made in the lab, so we know that these butterflies have ability to feed on those plants. And then here on the internal nodes of the butterfly tree, we have the inferred ancestral states with more than 90% posterior probability. And so then we can say that we infer that the ancestor of this group fed on an urticacea. You can't really know this just from the color here because plant families share the same color, but I know I can tell you that it's urticacea, this first plant family here. And then you can also see how there are some events here where there's more of a shift from one host to the other, but also some events of host range expansion, both in here and down here, where the butterflies kept using the ancestral host, but gained another host. So we call this an expansion and not a host switch. And then interestingly, there's also the small squares here, and they represent these potential interactions. But so here's a second example of a use of this model. And here we looked at a different group of butterflies. They're more specialized to their host plants. And here we use the two-state model. So we ignored potential hosts. And this is the same type of reconstruction, but that's not what I want to focus on in this example. So the same data can be represented as a network like this, and the butterfly tree is the same. So this matrix of interactions, it can also be represented like this network, and this tree is the same one as this one. It's just a little compressed. And then in this paper, we focused on this type of reconstruction where we ask what are the probability of interactions at different points in time, rather than just looking at the internal nodes. So doing this, we were able to reconstruct ancestral networks from 10, so this is the present day network, and then here is 10 million years ago, and then we went all the way back to 80 million years ago, around the time when about the time of the origin of this butterfly group. So to look into a little more detail, then we can see that so in all of these different figures that I'm going to show, we're always going to have the network and the tree is the butterfly tree. In each network, we have squares, and they are plant families, and the circles are butterfly species. The thickness of these edges in the network are proportional to their posterior probability. And the colors represent modules. So these modules are just groups of nodes. So butterflies and plants, they interact more with each other than with the rest of the network. And it's mainly to decrease the number of host plants in a way. So grouping host plants into these modules. Right, so what we can see here from looking at the network at 80 and 70 million years ago is that Fabaceae is the most likely ancestral host for this group, and followed by Caparaceae. But then when we look at the network at 60 million years ago, then we see that those are actually two separated modules, two separated groups. A blue one feeding on Caparaceae, and this red wine kind of color feeding on Fabaceae. And those are the more basal butterflies. And then if we compare with the network at 50 million years ago, then we can see this diversification of this group called Blerina that followed the colonization of Caparaceae, which is an example of a radiation followed by a host switch. And then there are also two other very important host colonizations in this clade. So at 40 million years ago, there were these two new modules that appeared, this yellow and the red one. So Laurentaceae and Centellaceae, these yellow module, they were colonized by a host shift, whereas Brassicaceae was colonized by both a host range expansion and the host shift in different parts of the network. So here you can see these two butterflies, they are only feeding on Brassicaceae. So I call this a host shift. While this butterfly here, it's still using the ancestral host, but also using the new host. So that's a host range expansion. And then it's even more clear when we look at the next time point, where you see when this clade diversifies, so you see that now you have several species that are interacting with both the ancestral and the new host. And at the network level, that makes these two modules, M2 and M3, be connected to each other. And that changes this overall structure of the network. And then from 20 million years ago forward, the network increases with butterfly diversification and colonization of new hosts. At the same time as recolonization of ancestral hosts connects new modules to older modules, and that further changes the network structure. And this process continues until the present, producing this very connected network. So conclusions from this study. So this is this approach of reconstructing ancestral host repertoires and using that analysis to reconstruct ancestral networks. That provides a means to test ideas about evolution of ecological networks with the new probabilistic representation that makes fuller use of the posterior distribution of ancestral states, because now instead of simply deciding on a threshold for a probability, a posterior probability, you can actually use that information in as the thickness of your edge, also the weight of your interactions. And we were, in our case, we were interested in reconstructing specific host shifts, host range expansions, and recolonizations that have shaped the Pieridae angiosperm network. So for those interested in the RevBase website, there is a tutorial on how to use the host repertoire model in RevBase. There's also an accompanying R package in GitHub, in my GitHub page. There's also code for pretty much all my papers. And then I also wanted to point out that this model has been used in other systems as well. So for example, I have used it to infer color pattern evolution in mimicry rings, biome evolution in beetles, and others have used it to study interactions between fish and parasitic mussels and between beetles and their host plants. So it's still quite early days, but it's quite exciting to see when others are also find this tool useful. So going way back to the beginning of my talk, I told you that I was interested in predicting new interactions, and then I didn't say anything more about this. So this is what I'm currently working on. So I'm implementing the host repertoire model in a new language, a new software for phylogenetics, which is called Tree People. Tree People is very much in early days. It's on development. We're just releasing our first release note. But the goal with this implementation is that we will be able to take an input like this one, where you have the interactions that you know, and then instead of saying that the other interactions, the white spaces, the question marks in your data set, instead of saying that those are no interactions, we can say, I don't know. And then by doing so, the goal is that the program will then output what's the probability of those interactions. So that would be a way for us to use both the information that we already have from closely related species, but also the information that we gain through modeling how these interactions evolve to then predict unknown interactions. And I'm quite excited about this, but it's still it's very much work in progress. Yeah. So with that, I would like to thank all my co-authors in the past many years, and thank you for listening.